Professor Vitkin is a physicist and biomedical engineer with specialization in medical physics and biomedical optics. He is a professor of medical biophysics and radiation oncology at the University of Toronto, senior scientist at the Ontario Cancer Institute, and clinical medical physicist at Princess Margaret Cancer Center. He has over 180 publications on uses of light in biomedicine, consults for several biophotonics and medical imaging companies, and is a topical editor of optics letters. He serves on many research grant panels, both nationally and internationally. He lectures worldwide, delivering seminars and summer school modules on biophotonics worldwide. And he is an active participant in the SBIE Visiting Lecturer and OSA Traveling Lecturer programs. He has won the Publication Impact Prize in Medical Physics through the Canadian Organization of Medical Physicists, and is a visiting professor of the Nizhny Novgorod State Medical Academy in Russia through the Leading Scientist Mega Grant Program. Dr. Vitkin is a board-certified medical physicist through the Canadian College of Physicists in Medicine, and is a fellow of the OSA and SBIE. And without further ado, Professor Vitkin.
Okay, great. I didn't realize you're actually going to read everything line by line. I better know what I write in those little blurbs. Now, the only problem is I'm trying to share a screen, but it says to me that host disabled participant screen sharing. So I guess we're going to have to cancel my lecture.
I already enabled it, so go ahead.
Okay.
Oh, is it better now?
Yes.
All right. No, it still says host disabled participant screen sharing. So still doesn't.
I just clicked it again. If you could please give it a try.
Right again? Yeah, okay. It's working now. Better. Thanks.
Okay, perfect.
All right. Let's see if this works. Is that visible to people?
Yes.
Yes.
Okay. So as advertised, and thanks very much for inviting me. And it's not nearly as good as physically being there, but I guess I'll just sit in my office and pretend that I'm speaking to Massachusetts crowd. I am going to talk about our optical imaging technologies, specifically optical coherence tomography, or even a slight variant of that optical coherence angiography, to image blood flow in the context of radiation therapy. So this hopefully will be of interest for both the medical physics, radiation oncology crowd, as well as the biophotonics crowd. And as advertised, I'm all based in Toronto, and I'm a medical physicist, and I do research in biomedical optics, and some of the two intersect as in the research I'll present today. Also, maybe in the interest of making this interactive, please feel free to just shout out questions or comments. You don't have to wait for 45 minutes or an hour at the end just to ask one or two questions or so. So make it more interesting if possible by participating. And logistically, can everyone see and hear me and see the slides okay? Everything's good? Okay, thanks. Because it's a little different presentation mode. You don't see as many people in the feedback loop as you do when you're just a participant. All right, so with that in mind, I'm going to talk what's the problem. So the problem is we're going to try to understand a little deeper what's happening in the radiated tissues and radiation therapy, and I'll tell you why there's still unanswered questions in that space. For context, radiotherapy, you may or may not know, is very widely used, tens of millions of treatments a year, essentially. In 50% of all cancer treatments, either alone or most likely in combination, use radiation therapy. It's very effective. It's totally, or if sometimes in the context of brachytherapy, minimally invasive, but it's so non-invasive that you really don't know what's happening all that well till weeks or sometimes even months after treatment, when you follow patient symptoms or do CT or MR scans and so forth. So the outcome is generally fairly predictable and well known, but it is somewhat variable. And certainly there are patient variations, interpatient variations, even with the same diagnosis and the same staging and so forth. So there's a lot of push in the radiation oncology community to meet this unmet clinical need. So can you monitor response on a treatment by treatment, patient by patient basis? and to offer essentially treatment, for example, mid-course corrections or modifications to the treatment in the context of so-called personalized medicine, or ART, which is adaptive radiotherapy. And there's different ways of adapting it, but basically feedback control or monitoring therapy progress to optimize it during the treatment delivery or between fractions or shortly after treatments and so forth. There's also emerging radiation therapy paradigms such as SBRT, which stands for stereotactic body radiotherapy. I'll get back to that a little later on. That brings in a whole set of new unknowns where the technology we're going to describe hopefully will shed some light on that. So to speak, there's also something called FLASH, which doesn't abbreviate for anything, but it just implies very high dose rate delivery of radiation and makes it even more needed, as it were, for, sorry, I'm just going to put on the, here. Makes it even more relevant in the context of feedback control and understanding what's actually happening on the kind of radiobiological microscopic level. So to address some of these things, we're going to try to zoom in with optical microscopy in vivo. So I'm going to talk about OCT, optical coherence imaging, or optical coherence tomography, and some of its properties, specifically its spectral properties, and how we use that to monitor microvasculature, blood flow primarily, and see if we can gain some insight on radiotherapy. So what is OCT? It's well known in the Boston area because that's roughly where it was invented. It's been used many times in many places, primarily in ophthalmology is its clinical use. But in research, it's a much wider use in ophthalmology. So it's basically an in vivo microscope. It enables to get you very high resolution images, but not very deep into the tissue. So the order of several millimeters at most. So we've been working in this space for quite a while, and I'll concentrate today on how we've adopted this technology specifically to monitor blood flow, microvasculature, kind of tissue functionality as opposed to tissue structure. And I've circled some of the things that are relevant to today's discussion. The video you see playing on the bottom is just one of the very first images or one of the first movies we've acquired. This is just a structural OCT movie playing in repeat mode of the beating heart of a tadpole, of a frog, sorry, beating heart of a tadpole, which is a miniature frog or frog baby. Frog embryo. It's called Xenopus lavis. This is a fairly well established and widely studied developmental biology model. Several interesting things to note. Let me just stop the video so we can concentrate on it. So the image is very speckly. We'll come back to that. You can see this. It's not just the pixelation, the resolution issue. It's got this bright and dark spots. And that corresponds to constructive and destructive interference of the infrared light that's used to image, to perform this OCT imaging. And that's the speckle business, this kind of iridescent, bright, dark, salt and pepper look to images is common to many so-called coherent or wave-like imaging techniques. For example, ultrasound is a well-known example. And generally, speckle is considered noise. It kind of degrades the features of the image. But in some cases, it actually carries some signal characteristics or something useful, and it will make use of that. Another thing to note is the scale. So this is microscopy, essentially. This is 500 microns or half a millimeter. So the whole field of view is not very deep. So laterally, it can be 2, 3, 4 millimeters, even more depending on the scanning system. But in depth, you're limited basically by tissue optics, although this is not biological, sorry, this is not mammalian tissue, this is amphibian tissue. But still, you're basically limited to about 2 millimeters in depth. And that's a limitation based on tissue optics, not on the good or the bad of your technologies. That's just the way it is. That's how tissue scattering works and degrades these properties of this image. And also, just out of interest, this is again beating heart of a tadpole, and you notice it has 3 chambers. The amphibians have 3 chambers as opposed to mammalians that have 4 chambers in the heart. Okay, so I won't belabor this. I'm sure anybody who's worked in the optics lab would know what a preclinical or benchtop system looks like. So there's lots of optics, lots of fiber optics detectors, lasers, light sources, LEDs, photo detectors, IQ cards, and so forth. So this is some of our preclinical systems that we use for small animal imaging, which I'll show you some of the results from. We also have a couple of similar systems that we've engineered and packaged on a card and taken to different clinical environments like radiation oncology environments and gastrointestinal endoscopic environments and so forth. But this is a benchtop system for preclinical small animal imaging. So what's so special about OCT, or not so special? What are its kind of defining characteristics? So probably its biggest selling point that it has really good contrast and very nice resolution. So the resolution of the order of microns, so kind of microscopic resolution, maybe a little worse, but depending on the system, can be close to that. So the order of, say, several microns to 10 of microns. The penetration depth is pretty poor though. So you have to choose preclinical or clinical scenarios where a couple of millimeters is where all the action is. So kind of mucosal layers of, different body orifices, or retinal layers when you do ocular imaging, skin layer, something like that. So it's not a technology to see through the entire animal or human or elephant or anything. It's not of the order of centimeters or tens of centimeters penetration depth or imaging depth. So it's somewhere in between these relatively invasive ex vivo technologies, which do amazing resolution, but can see almost no depth at all, just very thin tissues or very thin slices, and considerably high resolution that some of these other more conventional medical imaging modalities, which sacrifice resolution for much better penetration depth. And it's kind of obvious that this curve will have an inverse relationship. The greater the details you can see, the shallower you can see. But the slope is interesting. So it looks like if you line this up, roughly speaking, you get about 200 pixels or 200 resolution volumes into the tissue, into any material, is how far you can see. So for example, if you're looking at 1 millimeter scale resolution, you can see of the order of 20 to 30 centimeters into the tissue, and it scales accordingly. So it's just something interesting to notice. So OCT is somewhere in the middle, sometimes been called mesoscopic imaging, somewhere between macro and microscopic imaging. By the way, as I mentioned before, if anyone has any comments, questions, or concerns, don't wait till the very end. Just pipe in now and just shout out, or don't have to raise your hand because I can't really see that in the presentation mode so much. Okay, so before we get back into radiotherapy and how we adopt this technology for that, so what are the good, the bad, and the ugly of this approach? So again, I mentioned it's non-invasive, in vivo, cross-sectional, three-dimensional microscope, essentially, and it can be used in vivo relatively readily because most of the systems can be readily engineered through fiber optic access and endoscopic access and catheter access, etc. But it doesn't see very deep, although with micron scale resolution. That's on the structural side. And then we and others have adapted in many other ways to see kind of beyond structure. So I'll show you how we image microvasculature. There's a lot of work in many labs, including starting in our own lab, imaging tissue biomechanics, so elastography, analogous to what's used in MR, sometimes ultrasound elastography, just to look at the stiffness or elastic modulus of tissue. So the advantages are listed here. The bigger disadvantages are more perhaps the shallow penetration depth, which is advantage or disadvantage. It just depends on what clinical or preclinical application you gear this to. Limited field of view is not a big deal because the technology is developed to the point where you can have pretty large area scanning, contrast and resolution, clinical utility. Those are general considerations for any imaging modality for any task that you're interested in performing. So let's bring this back to radiotherapy and specifically to microvascular effects of radiotherapy. Why do we want to image tissue microcirculation, microvasculature, blood flow? is because it's well known, and I'm sure whoever's looked at any slides or oncology knows that the normal architecture of the microvascular tree is much more ordered and much more fractal-like, much more space filling. There's an ordered progression from arteries to arterioles and back to the venules and to veins and connected through this capillary network, which is thin enough to essentially allow one red blood cell at a time to squeeze through, or at least one. Quite often they're lined up in this Rollo or other types of complicated flow patterns, but it's wide enough to basically allow one thickness of deformable red blood cell. More importantly, when you look at tumors microvasculature, not only does that architecture look very different, it's more chaotic and unpredictable, but perhaps in the context of therapy, it's much more unresponsive. It's much more or much less well regulated. So both its structure and its functionality are very different. This is exploited in angiogenic or anti-angiogenic therapies in oncology, where you're not targeting the cancer cells, but you're targeting the differences in the blood supply between normal and pathologic tissue. The problem is that these are either electron micrographs, electron microscopy micrographs, or micro CT, which is X-ray tomography, but very large doses and many, many projections. So both of these are not very, in fact, totally unsuitable, or sometimes very rather unsuitable for in vivo preclinical or even clinical imaging. So the challenge is, how would you obtain comparable images in living tissues? This is a plastic cast of dead tissue, because there's also plastic X-ray contrast agent injected into here. So that's where OCT comes in, or that's one possibility where OCT can be used. So we developed this technology about 10 years ago. And that's shown in the bottom right, and I'll explain how that works. So how we did this, and it doesn't have to be implemented exactly this way, but the way we implemented back then was to use something that helped us, a window chamber. So this is a piece of glass that you can kind of surgically insert or surgically implant into the brain, or into, in this case, skin fold, dorsal skin of a mouse. This is an immunocompromised mouse, so that we can seed this with human, for example, tumor cells, and monitor how tumors develop, and through this window do optical imaging of the collagen structure, the microvasculature, any other considerations, that just the cell growth itself. So it kind of gives us an in vivo model of pretty well controlled disease progression and treatment response monitoring. So we can do photodynamic therapy or chemotherapy, or in my case that I'm going to present, radiation therapy on the tumor as well as the normal tissue within this window chamber, and monitor what's going on in time. So this was how we developed this based on speckle variants. I'll show that in the next slide. And since nobody, including ourselves, believed that this was real, because it looked a little too good to be true, we did confirmatory microscopy, where this is with a contrast agent, in fact, the injection. So this is fluorescent contrast agent, fixodextran, injected into the vasculature, high molecular weight, so it stays confined to the vessels, and we see where the vessels are. And this is a maximum intensity projection of OCT speckle variants to monitor in vivo tissue microvasculature. So how does that work? So you create a whole bunch of relatively boring structural cross-sectional OCT images. And then what you basically do is you compare them in time. So in the same location in space, you create these speckly cross-sectional images, and then you mathematically examine every voxel or every pixel of these two-dimensional images, and monitor if the speckle structure, however complicated it is in that pixel, changes in time or not. And our thinking was that if you have quote unquote solid tissue, like relatively fixed, then this kind of temporal spatial filtering will reveal a similar speckle structure as you go across these frames. But if you have low viscosity flowing or even Brownian motion scatterers that are less rigid in space and therefore in time, then you'll have variation in the speckle. So that's why we call that speckle variance. And so basically the contrast is defined through this very simple equation. We just look across the frames to see if the pixel is changing its intensity or not. And if it is, we attribute that to liquid tissue, i.e. blood. And if it's not, we attribute that to solid tissue, and that's basically not our blood signal. So based on this approach, and we've done a fair amount of work contrasting this with Doppler, the pros and cons of this and so forth, we can get pretty nice images, which are, in our case, although it doesn't have to be flow independent. So it's important to note we're actually not monitoring flow per se. We're just monitoring the architecture of the pipes that contain the blood. We're not making a comment right now whether that blood is flowing fast or slow, or maybe it's even stagnant, or we're only detecting Brownian motion. But that's dependent on the timing between frames and the number of frames and the density of scanning within the frame to obtain this two-dimensional image and so forth. But we can get back to that if anyone has questions. So with that contrast idea in mind, we can process our basically stacks of two-dimensional images to create three-dimensional pictures like what is shown here. So what is shown here is basically a speckle variance contrast, and it's depth encoded. So the superficial speckles are yellow, and the deeper ones are darker colors, red. It's only 0.5 millimeter, so it's a very thin tumor. And this is a three-dimensional vascular map. Still has some artifacts. I'm sure you can appreciate these are not real blood vessels. Why that directly, that's kind of our imaging direction and so forth. But at the same time, this does give you amazing in vivo indication of microvascular structure. And this is just a blow up of that. In addition to that, those pictures, and this is what normal vasculature looks like in the skin. And this is depth encoded three-dimensional tumorous vasculature. You can begin to quantify some numbers. So you're not just showing pictures. That's pretty obvious, the difference in the microarchitecture of the blood between normal and tumorous tissue. You can begin to quantify some numbers like fractal dimension and fraction of small versus large vessels, depending on how you define that threshold, and average branching ratios, tortuosity, and so forth. And we're in the process of examining some of these metrics to quantify these images. Right. So with that in mind, let's go back to how we're going to tackle and shed some light, quote unquote, on radiotherapy and why it's even important to look at vascular response. So the context is the following, that in conventional radiotherapy, which is fairly low doses of the order of 1 to 2, well, typically 2 gray per day, spread out or fractionated over several weeks, like 5 or 6 weeks is a typical treatment. The prevailing thinking is that the vascular response is not really that important. And most of the tumor death and tumor kill is through the cellular actions, through the double strand breaks of the DNA and so forth, kind of classical radiobiology. However, when the doses become large, of the order of 10 gray or more, and that's a pretty soft number, the vascular involvement seems to become more important. And I mentioned briefly in the beginning SBRT, stereotactic radiotherapy. That's an emerging treatment paradigm in radiation oncology, where you do actually give large doses, but much fewer fractions. So instead of 2 grays times 25 or 30 over 5 or 6 weeks, you give, for example, 7 or say 12 grays times 4. There's a lot of radiobiological equivalence as to how you equate those doses over time and etc. But the bottom line is there are a lot of pushes towards higher dose treatments and fewer fractions for economic and radiobiological reasons. So the vascular context is becoming more important in radiation oncology. This is what I said is generally trends, but it's not very conclusive because the data is very incomplete and inconsistent, especially in vivo data. And there's still questions as to, even though the vasculature may be more involved or more changing, is it a passenger or a driver? What's its actual role in the eventual tumor response. Those are some unanswered questions. And again, I just mentioned the relevance that there's an emergent trend in oncology that you have fewer fractions and more dose per fraction. And this flash, this extremely high dose rate of the order of seconds, radiation delivery in some ways is the ultimate, and this hyperfractionated therapy just blasts everything very, very quickly. It's not really known. A lot of people are studying it or getting on this kind of trend, and there's a lot of unknowns in this space as well. As well as I know in the Massachusetts area, you guys have a proton radiotherapy, and some other people have not only protons, but heavy ion radiotherapy. And there's also some reports of interesting effects on microvasculature. I think I heard a question. Somebody had a question or comments? No? Actually, since I stopped anyways, does anyone have any questions or comments so far? All good? Okay, proceed uncorrected. All right. So it would be nice for us to then look at the dynamics of post-radiotherapy vessel response. It's been reported that that's one of the drivers of failure of radiotherapy, like tumor regrowth and relapse. So can we image, understand, and potentially target? This is just showing some of the literature associated with these higher dose rate effects, which seem to be fairly effective, which is good, but the clinical and even kind of in radiobiological insight is missing, because we need to understand those mechanisms a little better. So there's another model, or actually one of the several models in the literature, also coming from the Massachusetts area. This is Rakesh Jain and the Steele's lab in Boston, who kind of try to understand the effects, propose a model based on somewhat incomplete literature that's been reported of experimental effects and how tumor size, and this is really in this paper, a surrogate of microvasculature, how that changes with time following a radiation delivery. So this is a kind of a fake experiment with a single dose. We never use single dose in radiation oncology because it always fails unless you give it to the point where killing the tumor, but by that level you would have killed the patient. So that's why we always fractionate. So you can see that no matter what happens, things relapse and fail, at least according to this model. So, but anyways, the temporal dynamics are quite interesting. The tumor size and tumor vasculature grows unabated, and you arrest that with a dose of radiation. Again, this in the model is of the order of 20 gray. And then the tumor begins to regress, the vasculature decreases, and then things begin to take off again and fail. So he proposes different mechanisms for different regimes in this temporal response. But his paper actually concludes, or somewhere in the conclusion, there's a quote saying most models of post-radiation therapy vascular dynamics should be validated using modern technologies. So we thought we can make a dent in that space because we have some of these modern technologies. So again, this is the platform we're using. I mentioned the window chamber, this piece of glass you can implant, and then you can implant a tumor cells in there, watch it grow, watch its vasculature develop, connective tissue, and so forth. Then you can administer well-controlled doses through this microradiator. We know our medical physics, so we can do chromic field symmetry and know what doses we're given, what field sizes, and so forth. And then monitor in time what's happening. So here is a brief preview of what we can see. So this is this microvascular maps on a very short time scale. So this is following, I believe, 10 gray. So pretty modest dose as far as vascular effects are concerned, but something we can definitely see. And this is the vasculature of the tumor, again, three-dimensional prior to irradiation. So about 2 weeks after following tumor implantation into this window chamber. And you can see some transient shutdown of the vasculature. This is of the order of minutes or tens of minutes following this dose delivery. The fraction, the percentage is just percentage relative to the vascular volume fraction. Arbitrarily defines 100% prior to radiation. We can carry this much further though and do this work. And this is a very brief one graph summary of about 2 years worth of work and accumulation of statistics and image processing and so forth. So this is of the order of almost 2 months. So 7 weeks following, 8 weeks following window chamber and tumor implantation, 6 weeks following radiation. As a function of dose, so this is 10 gray, 20 gray, 30 gray. So there's a lot of features in these curves, but roughly speaking, you can see it kind of lines up relatively interestingly and kind of corresponds to the general trend that was proposed in literature. So unpacking that a little bit further, there's a lot of work we've done, and I wouldn't say it's conclusive. There's just different choices of what type of preclinical model, what host you choose, what tumor cell lines you choose. Do you go with tumor cell lines or tumor xenografts, patient-derived xenografts perhaps. But this is just showing you what it looks like in the absence of radiation, just when you implant tumor cells into this window chamber, and you can watch it grow over up to 2-3 weeks or longer if you want. And just for reference, here's what normal skin microvasculature looks like. So much more ordered and actually generally spatially oriented. There's a preferred direction as to how the capillaries line up, at least in mouse skin. Right, so I'm just going to show some results for pancreatic cancer. So this is what the pictures look like if you follow one mouse. Again, this is a lot of experimental work, a lot of image processing, but this is what the features look like up to 7 weeks following radiation. 20 gray, I should have actually indicated field size, but it's of the order of 4 millimeters across, so it's more or less encompassing the whole tumor. It's a little smaller than the window chamber, so we can also monitor the normal, quote unquote, normal tissue / unirradiated tissue response in the same window chamber. And that's actually quite important because we want to see the difference. I won't show that data here, but we have some analysis along those lines. So basically, not only unirradiated tissue over the same time course, but tissue adjacent to the primary beam tissue in the penumbra, although the penumbra is fairly minimal here, but basically adjacent, quote unquote, normal, quote unquote, unirradiated tissue. So we can have some normalization possibilities. So this is the same data as I showed you very briefly with a little more error bars and a little more detail. So this is response of the microvasculature just to single dose, increasing dose. We also monitor, this is all that in the same plot. I've showed you the model. We've also monitored some other metrics associated with the tumor growth and tumor response to radiation therapy. So this is our OCT metric again shown. We've also looked at the physical size of the tumor, like just through caliper measurements, how big is the tumor in 3 dimensions. We also look at the viability of the cellular compartment of the tumor, how live is it? So we can begin to now kind of put together, and we're still in the process of doing it, some radiobiological models as to what's happening in the whole response space. So let me skip that. Been through that before. One thing we're totally aware of is that we're really underselling our data. So all those rich 3-dimensional vascular networks that we acquired at OCT for the purposes of the paper I just showed you, would just transform that to a single number that talks about vascular volume density. There's many more spatial features that we're trying to extract now. We're also trying to attempt to use kind of artificial intelligence machine learning to do some of this predictive modeling in time, as opposed to reducing the work of the 3-dimensional images just to a single number. Haven't actually gotten very far in this, even though I've been trying, because even though the data size, we don't have that many images. You know, we've done this maybe with 100 mice. It's, you know, each mouse has maybe 15 or 20 time points over 6 weeks, and at each time point there's humongous amount of data, you know, 3-dimensional data cubes, but it's not hundreds of thousands of images. So we have to make better use of the images we have to be able to deal with the large numbers required for kind of machine learning classification algorithms, prediction algorithms. So we need to continue better in this space. Let me tell you some, if time permits, and I guess we do have about 20 more minutes, some other stuff that I've been working on. I'll also show you some of how we've taken what I showed you to the clinic as well. One other thing we noticed during our kind of OCT image processing and image acquisition is that we are seeing some structures that don't seem to correspond to blood vessels. And so we started looking around and seeing what people have done to monitor other liquids or fluids in tissue. And there's been a fair amount of work on OCT for detecting transparent tissues. So lymph is a prime example. Also biologically very important, maybe not, well, it's not a competition, but maybe not as important as blood, but it's another crucial component of circulating fluid in the body. From an optics point of view, it's a little harder to see because you don't see it. To a first approximation, it's transparent. It's kind of like saliva or tear ducts or urine or cerebrospinal fluid. It's not very optically active. It's not very exciting. It doesn't scatter or reflect light so much. It might have some color, but we don't detect that so much in OCT. So a lot of the previous approaches for this based on OCT have basically used the absence of signal, for example, as you see here, basically a hole or lack of reflectivity as a means of detecting microvasculature, and that can be very effective. But it's very tricky in the low signal in the regions close to noise, because almost by definition, noise is where you also have no signal. So it's very difficult to tell these holes from true noise, especially if you go a little deeper into the tissue. So we try to look at a little more into the speckle statistics of this, and we found that we can find an approach based on probability density distributions, whereby we know what they should look like for pure noise. It's like a Rayleigh distribution if we look at speckle intensities over a region. If it's very far from Rayleigh, we know that it's either blood or other tissue, something very, very scattering. So we know it's not this section we're interested in. But if it's somewhere in the sweet spot, what it begins to look like noise, but not quite the same as noise. So we basically do this analysis with regions of interest across the entire structural field of view, and where it looks like the same speckle distribution as noise, but not quite the same. We've been able to show that that actually corresponds to lymphatics. And the advantage of this approach as to compared to looking for holes or absence of signal is that you're much less prone to noise effects. So this gives us an idea of, or possibility of detecting this lymphatic microvasculature in low SNR regimes. And as soon as you got to any depth in OCT imaging, you're always in potentially low SNR regimes. So I won't go into the details of how we do that, but it's slightly different signal processing. We're trying to see how that differs between normal and tumor tissue, how it's relevant in oncology. We haven't answered that yet, and the literature is kind of confusing on that as well, and what its relevance to radiation therapy, which was the theme of my talk. One other interesting thing to note is that we also noticed we can see other semi-transparent structures through this approach. And we didn't know what they were because they didn't seem to correspond to lymphatics when compared to histology. But with a little more advanced staining and immunohistochemical support from some of our pathology colleagues, we realized we're also seeing nerves, because nerves and associated collagen fibers around the nerves, for example, white myelin sheath in the brain, although this is in the skin, are also kind of semi-transparent pipes or architectural features that we can see with this. So we now have a means, I don't think as robust as what we can do with blood vessels, just because the signal characteristics are different. But if pushed further, I think we can detect lymphatics and nerves in live tissues as well. And so this is shown for normal tissues. This is corresponding results for tumorous tissues. So again, whether that's relevant in radiation oncology in the context of radiation response monitoring remains to be seen. We don't yet know One other thing I should mention, this is just the last 2-3 weeks, we've been trying to link this up to a more clinically translatable approach. And that's, for example, MR imaging is a primary example. It's widely used in radiation oncology, and there's things like MR Linux, where you can actually have technology of both of those things in the same hybrid instrument. But MR, as I mentioned, is, while clinically applicable, doesn't have the spatial resolution that we do with OCT. So our idea was, could we do this OCT and preclinical MR in the same living animal and compare the different metrics and use one to inform another? So you can see this is just an optical image, not OCT, but just visual. How much more pixelated and poorly resolved the MR is, which is why I mentioned that it's not really imaging tumor microvasculature. I call it kind of macrovasculature, or like the overall response. However, MR can be pretty quantitative. And there's a lot of already derived parameters. People talk about K-trans and fractional volume of extra-rated contrast agent, gadolinium contrast agents. You can get semi-quantitative, quantitative, wash in, wash out. There's compartmental models. There's a lot of work in this space that's available for us to draw upon. So right now, and this is just, again, last 2 weeks, we're trying to link up some of these metrics and these kind of poor resolution images, but can be quantified relatively readily, to something that we can just extract from OCT, from microvascular OCT. And the idea, again, is to kind of inform these metrics kind of in the background a little more with the microvascular effects that we are seeing in vivo with OCT. And if we can form just decent correlations and understand what's going on, we could potentially link up some of these measurements into the clinical MR translation space, to be seen whether that's to be successful or not. We have, I mentioned briefly, not only done preclinical OCT imaging, but done some clinical work with patients in radiation oncology space. So I'll show you 1 or 2 examples. This is with collaborators in what was mentioned in Nizhny Novgorod State Medical Academy in Russia. So we've implemented some of the same approaches of microvascular imaging, designed some oral probes, and we're looking at head and neck radiotherapy patients to look at the problem of so-called mucositis, one of the common side effects of head and neck irradiation. This is a radiation oncology treatment plan, radiation therapy treatment plan, where one site had pathology, and therefore most of the dose is concentrated on this right side, patient's right. And this is some of our vascular metrics, vascular density, and a fraction of small vessels that we can do from from image analysis of these types of volumes, vascular volumes, obtained in vivo from these patients. And the arrow shows when there's first clinical manifestations of mucositis, this debilitating side effects that happen during a lot of upper oral cavity head and neck irradiation, where you lose taste, you can't swallow, there's very little saliva, and so forth. So we can see vascular changes considerably earlier, not surprisingly, compared to the contralateral control, than the clinical symptom manifestation. And there's some hope that there are mitigating strategies, both in dose modifications or in some post-dose delivered mucositis management, that we can possibly flatten out these curves and avoid the onset of mucositis. So that's to be determined. But at least we show that we can see the differences early. So just to kind of summarize briefly, we've done it and continuing to do a fair amount of preliminary work. Probably the current pushes now is better quantification of the images we have, progression to a multi-fraction regime as opposed to a single dose, what I showed you, and this linkages to MR. I think that's where we are mostly at this stage. And linking this up to this SBRT regime, high dose per fraction, and potentially this flash, if and when it becomes more readily available for us to study. So to wrap up, on the physics and engineering techie side, we seem to be relatively well positioned. We have a decent platform for, I think, functional tissue imaging, both preclinical and a little bit towards clinical. We do need to add some blood flow information, and we are working on that. Analyze our results better than just that single vascular volume number I mentioned before. We do have lymphatics and nerves imaging. We need to improve that and also see if that has any relevance or any play in radiation oncology. We simply don't know that yet. And ideally correlate with the clinical imaging approaches, for example, MRI. On the bio side, there's a lot of unanswered questions. So we're showing nice pictures and decent images, but what does it mean? We're not quite sure. So we'll probably investigate if there's really a threshold dose for vascular involvement, because we do have solid technology for that. We do know that small vessels respond differently than large vessels, so we might play on that. We will be doing fractionation studies, so not just single dose, but higher dose per fraction, a few fractions, potentially doing this really high dose rate experiments as well. All of the metrics and differences we see, we still need to make sure that they make any difference in terms of eventual patient outcome. Just because we see differences that may or may not translate into tumor control several weeks or several months later. And we haven't really touched on this at all, but do we ever use this information to feedback on treatments? Do we alter treatment delivery based on the trajectory of how, for example, the vasculature changes after 2 fractions? Do we alter the remaining 3 fractions? And do we stay the course? Or do we know that it's going to fail or you'll have unacceptable side effects and so forth? Haven't really answered any of those questions, but at least we are, I think, in a reasonable position to begin to at least intelligently pose those questions. All right, so on that cheerful note, I'll thank all the people and granting agencies and you for your attention. Okay.
I've got a question, if that's all right.
Absolutely.
So I know you haven't gotten into, it looks like you haven't gotten into the dose rate with Flash yet, but I know one of the surprises is that some of the toxicity isn't as high as was expected when we started talking about these dose rates originally. Do you think that has something to do with the vascular response, or do you have any explanation for why that might be?
No, and I don't think anyone else does. I mean, I would certainly think, tissue microenvironment is a major determining factor in this. It's not just cellular kill, but I think that's a guess. And it's, if you look in the literature, it's the same level of guessing what it's likely to be, what it's likely not. But I mean, almost like SBRT, people are pleasantly surprised, and even more so that this seems to be effective in terms of, adjacent normal tissue sparing. That's great. But I think to fully take advantage of that would be good to understand it. So, because it's a long ways of saying, no, I don't know what's causing it. But I would think that some of the functionalities that we can explore and the others can explore too would tend to explain it, or would have a role in explaining that better. My guess is it's not just DNA damage to the cellular compartment, because that shouldn't really depend on dose rate, I would think. I don't know. I mean, might be radiation oncology experts in the audience who could weigh in on this conversation.
Well, thank you very much.
Oh, you're more than welcome. Sorry, I wasn't able to say yes, no.
It's okay. I mean, I didn't expect you to have all the answers right now, but
Yeah.
It looks like hopefully we can get them eventually.
Yes.
So, the Flash therapy seems to defy the 4 R's of radiation biology. And one of the things that in addition to what you mentioned is that it's just don't have enough experience with it, is that at these doses and those rates that Flash delivers, you might expect some late effects. And so that is also something that would point to not just the involvement of DMA double-strand break, but all kinds of other additional effects, including vasculature.
When you say late effect, do you mean of the order of like half a year to a year, or?
Well, probably less than that. Probably in these doses and those rates, probably half a year, something like it. Yes.
Several months, yeah.
7 months.
No, and people are starting to study these, but I think the jury is still out as to what these effects are, and certainly what the explanation of these effects, whatever they are, what is the explanation? I think it remains to be seen.
In your experience, when you talk about flash, do you mean electron flash or photon flash?
I probably mean electron flash because that's like more technologically accessible. But in reality, I don't really mean anything because we haven't done it yet.
Right.
Yeah. But do you guys have capability for doing X-ray flash or photon flash?
Not yet. We are about to collaborate with MD Anderson to test some things out, actually a detector.
On electrons or?
On electrons. So they have just installed an electron flash unit in the last month or so.
Does anyone actually, this is for my own edification, I couldn't find the abbreviation for it. Does flash stand for anything?
I don't think so. You are right in saying that flash is probably just referring to the very short period of time and very high dose rate.
Gotcha. Yes.
I'm not aware of any, so not aware of the fact that this might be an acronym. I think it's just a word.
Fast lightning, or I don't know, something.
Right.
Yeah.
Fast lightning, I don't know.
That's right. We could make it up during the seminar.
Right.
Contribution to science. Okay. Any other comments or suggestions or questions or discussion points?
I have a question concerning oral mucositis. So how early can you see the changes, and how many patients did you look at? So
okay, yeah. So this was, let me just go back to the slides if you want. The number of patients was pretty modest. I think like 27 or of the order of 30 patients. The changes were seen pretty robustly and pretty early. So I would say certainly within the first 10 days, which is like 4 to 5 fractions out of a 20 or 30 fraction regime, we can see changes, and they're typically seen of the cumulative dose 15 to 20 gray is when the patient, the manifestation are clinically detectable. So we can see maybe twice as early as what the clinical symptoms are.
right, and these curves, that's just like one patient, or?
Yeah, I think these are specific, right? So we have summary tables in this paper, but these are, yes, specific to individual patients, because for each curve we also showed the dose distribution for that particular patient. They differ slightly.
So there is increase in vasculature density, and it looks like it's pretty big increase, right?
Yeah, it was pretty significant. Yeah, it was certainly detectable. It was, I don't think out of the 29 or 30 patients we monitored, of the ones that not everybody reported mucositis, of the ones that did, I don't think we saw any false negatives. In other words, we didn't miss any. I think there was one case where we saw an increase and the patient did not report mucositis-like symptoms. So it's definitely not perfect, but it showed kind of feasibility.
Okay, and you didn't monitor any oxygenation or?
No.
Okay. Because like I'm looking from the point of view of this low-level light therapies and their dosimetry, if there is significant increase in vascularization, there should be probably adjustment of treatment protocol.
Sorry, which low level, which therapy you mean?
Low level, it's photobiomodulation therapy.
Okay.
They're now trying to use it to prevent oral mucositis.
So this is photobiomodulation and all that work? Yeah, that's tough. I think it's a good idea, and I think there are definitely microvascular effects. Whether they're kind of unequivocally related to LITT. Yeah, but you're right. Yeah, anything to do with like metabolism, tumor microenvironment, that's probably called into question, like becomes important when you do like light photo biomodulation.
Because that's a significant increase. And I have other questions which are kind of more research oriented. This glioblastoma images that you've shown, is it like cells injected into under skin of the mice, or is it in the brain?
No, this is, I didn't show any brain stuff. So this is all mostly dorsal window chamber. Now we're doing abdominal and thigh. And quite often what we've recently found that in this window chamber thing, injection of tumor cells is not optimal. Just practically, the tumor doesn't grow very deep into like the depth dimension. It becomes like stays more of a pancake, which is not that representative of three-dimensional volumes. And also the animal doesn't tend to survive that long, like 2-3 weeks. So what we do now is we grow tumors and then implant it, we grow the tumors into the dorsal or whatever site we do, and then we implant the window chamber when the tumor is already well kind of integrated into the host, just a practical thing. So it's less of tumor cell injection and more of a window chamber once the tumor is already grown or is growing. But all of the stuff I showed you, none of it is in the brain.
Right, okay, but this glioblastoma cells, and like, how do you, can you say anything about what was like the stage of the tumor? Because you have shown a lot of like very serious vascularization there.
Yes, so most of the stuff I showed was pancreatic tumor, but yeah, I showed some examples of those glioblastoma cells. I mean, this is immunocompromised mouse, so you can transplant human cell lines into that. I don't know how to answer the stage question because
there's probably no answer because if it's just cells.
Yeah, so it's just cells, yeah.
Okay, thank you.
Oh, not at all. Thanks, Anna.
Hello, I have a question on the same topic for the window chamber experiments.
Yep.
So if you were implanting the tumor, even if the tumor was smaller than the window, can you make a comparison on tumor vasculature to normal vasculature, or would that not fit since the tumor is not grown in the mouse?
Oh,
I was thinking,
sure, you can do that.
quantify the contrast of tumor versus normal.
Yes, yeah, no, I showed, I guess I didn't do a very good job, but I showed several examples. For example, here, if you just want to zoom in, this is normal skin vasculature in the window chamber, like with no tumor implantation. And this is, again, depth encoded tumor vasculature. Is that what you're asking?
But different mice?
These are different mice. You could do, I don't probably have this image, sorry. You can do
I'm just thinking, could you use that to quantify how much the tumor will, how different the tumor will look than the surrounding normal vasculature?
Oh, absolutely. Yes. Yeah. You can do that. We've even done that in the context of detecting tumor edges, and you can do it in the same window chamber. You just have to make sure you're letting, I think I do show that. Yeah. Okay. That's probably even from the same window chamber, but you can certainly do what you're saying. You could easily see where the feeding vessels and the more normal kind of ordered and somewhat straighted microvascular architecture is relative to this kind of random looking ball of spaghetti that begins to resemble tumor vasculature, if I understand your question correctly.
Mm-hmm. Yes, and this is the perfect image for my next question. So for these quantitative metrics, to what order do they vary between normal and glioblastoma? I see the example here, but have you decided so far maybe what's the ideal metric, or are you still grabbing as many as you can to see what?
Yeah, good question. So in my opinion, the differences are pretty stark in just about every metric you choose. But we haven't really concentrated on tumor to normal delineation because, in my opinion, it's not that important. I was much more interested in temporal evolution of these differences and actually attributing that temporal evolution to the intervention as opposed to just natural disease progression over time, because none of it is static. But certainly things like tortuosity and fractal dimensions seem to pretty robustly pick up the difference. In my opinion, you don't really need all this fancy image processing. You can just look by eye, and it's not rocket science to see that this looks very different. It is, having said that, when you look at many tumor images, they're all so different like that. And I don't have one, but I can probably, if you're interested, find an image exactly what I mean. Whereas the normal vasculature is much similar, like one mouse next or one region next to another region, the tumor variation in the tumor chaotic structure is very large. So some you see more space filling, some less space filling, some more larger preponderance of larger vessels and smaller preponderance. So it's the variation in the tumor is much larger, intratumoral variation, than the variation in normal tissue. But like tumor to normal, that's pretty unequivocal.
Mm-hmm. Great, thank you.
You're welcome.
And I have one more question. You mentioned that you also use fluorescence.
Yeah, we just use it sometimes to understand what's going on and to check for leakiness of the vessels to see if we can see interstitial fluid. Yeah, so.
Is it endogenous or exogenous? What
No, this is exogenous. So we inject fixidextran into the vasculature and we play around with its molecular weight. So if we want to just have it, stay in the vessels and to see the vessels, we would use something like this, which would be high molecular weight fixidextran. If you want to see if we can see interstitial fluid and how things leak out, etc., we would do the same experiment with low molecular weight fixidextran and watch it, initially in the vessels with confocal microscopy and then out of the vessels and see if we can correlate that with what we're seeing on microvascular or interstitial OCT.
Thank you.
Yeah, you're welcome. Okay.
I have a couple of questions. Thank you for a very interesting presentation. You talked about the temporal evolution, and it seems like there's a lot of potential for the technology. I was wondering if you think that the tumor microvascular response is better suited for treatment verification after the full course of the radiotherapy, or if it could even be used for adaptive planning in between fractions, because you showed some images where there seems to be a pretty quick response, just like 30 to 60 minutes even after delivery.
Yeah. No, that's a great question. Thanks. I don't think it should supplant, tumor volume measurements or cellular kill fraction measurements or anything like that. I think it forms, it's another information channel. We haven't done the actual work to, for example, with reference to those early responses to see if they predictive it. Just because we can measure them and we can quantify them, it could be non-specific, swelling or something, or reddening of the tissue following radiation. And if that happens to everybody, why that wouldn't be a good predictor of who will fail in 6 weeks and who will regrow or who will regress in 6 weeks. So I think there's, we certainly are keen on exploiting the vascular response and seeing its role in predictability of outcome, but we're not ready to state, this is the metric we should measure, and if we measure it well, this is our, radiation effectiveness dosimeter. But we do need to do more work in that direction in terms of correlating with outcome and seeing how robust these metrics are for temporal prediction of where we end up and so forth. And of course, the idea in the long term would be great that after, 50% into your therapy, say after 10 fractions of conventional radiotherapy, after 2 fractions of SBRT, you would have some temporal trajectory as to how the normal tissue is going to respond and how the tumor's tissue is going to respond, and then therefore proceed as planned and don't change anything, or you're wasting your time because tumor's not responding, so it's just not happening, or it's responding, but the surrounding normal tissue will have side effects which will negate the benefit of the treatment or something like that. But that's just hypothetical. We're not there yet.
Yeah, no, that makes sense. And then the other thing I was wondering is, I think you touched upon this towards the end of your presentation, but do you think like radiomics or machine learning has any promise to generalize the results that you found to other in vivo tumor models? Like
I would like to think so. Yes, I think we need to explore that parameter space much more expertly and aggressively. Yes, so I think there's way more information in just even for a given tumor, I'm just like even in that, than the eye can see, and then we can do with our simple, vascular volume density or fractal dimension metric and stuff. The temporal and the spatial variation of the three-dimensional tumor response, I'm sure, has some insightful predictive information. But I think we're too dumb to get it right now just by looking at this images or just analyzing the very first order changes that we can see.
Yeah, if there are additional parameters. Thank you.
That's, again, my supposition. I guess the proof will be in the pudding if we can pull something out.
Thank you.
Okay. Great. Well, there seems to be some interest. People are asking some questions, so that's a good thing.
I have some more questions about the translation results that you showed, the clinical study for oral mucositis.
Yeah.
So I wanted to ask about kind of your vision. What would be the first sites to put this technology in the clinic? I understand because of access, that was a starting point, but does that have to do also with what radiobiology models are available for specific cancers, or how would you go about choosing the first application in the clinic?
I mean, if it's just OCT based without the MR linkages that I was showing towards the end, then I would certainly think that anything in mucosal layer and body orifices and kind of endoscopic catheter access would be the first order of business. So from that point of view, like upper GI, like esophagus and mouth, is a very nice target, and there are a lot of complications from radiotherapy as there are in any mucosal layers, and rapidly dividing cells that line various body orifices. And so the engineering of those probes can be much more sophisticated than what I showed in that picture. There are a lot of advanced technologies for catheter-based OCT, intravascular OCT, flexible access, like flexible endoscopes and catheters. Whether you could easily implement this speckle variance approach, because this requires kind of repeated scanning in the same location, spatial scanning patterns, that would be a more challenging engineering design question. Not unsolvable one, we haven't pursued it. But to go back to your original question, I think the upper GI and the bronchial tree is relatively accessible with flexible catheter access, and there are radiobiological reasons to look at those and clinical reasons to look at those. So I would think upper GI or lower GI for that matter, upper bronchial tree.
Would you need to, so when you said to get back to make the same measurement, would you need to have some landmarks? Would those come?
Yeah, I actually don't think so. I don't think, and we talked a lot about that during that clinical study. I think the advantage of this approach is that the metrics, whatever we're extracting on the fields of view, et cetera, should be representative of that region. If we're reliant on going back to the same, 3 cubic millimeters that we want to image for several days or weeks, I don't think that's too easy. And B, I think it's unnecessary. I think it's, we're not basing our numbers on a single vessel. Has that responded or not? If you didn't image it on Friday, where you may image it on Wednesday, your conclusions change fully. I don't think that's a good way to go. I think what we're imaging here is kind of microvascular response on regions of, the order of centimeters or so, at least an area. So I don't think there's a need to go to the same micro region. Yeah, pretty sure that's the case. So of course, if you go somewhere else and you measure the inner ear, and then the next day you measure the nasal cavity, of course that's different. But if you're always staying in the nasal cavity, the exact quadrant you're in, down to a millimeter, I don't think matters. It shouldn't matter. If it does, then we didn't do this well, and this is not a good metric.
Mm-hmm. And how long would, or at the moment, how long does each imaging session take?
So that varies, of the order of few seconds, like sometimes tens of seconds, to if the patient is able to, stay still, which is almost never the case, and we do have immobilization devices, etc., can extend up to tens of seconds. In the preclinical environment, we're a little more flexible, so we can image it on a large field of view with really beautiful maps. We can extend that to, say, several minutes. But that's in that order of magnitude. Obviously, with patients, and even it's kind of funny because you try it on yourself with volunteers, and you're interested, you're vested, and supposedly relatively healthy. You can stand still better and work with the people to make the images better. When you go into patients who are quite often elderly and not that interested and apprehensive as they should be, it becomes that much more difficult, and therefore you have to image faster and acquire the imaging faster and be cognizant of that.
Mm-hmm. Great, thank you.
You're welcome. Okay, is there more questions or comments?
So if nobody has any more questions, we would like to thank you, Alex, for coming here. And this is truly a very interesting discussion which generated a fair amount of interest among students.
Excellent. I'm glad, and please get in touch with me. I'm sure Anna or Tyler has my contact info and would love to talk more. If anyone, for example, specifically is interested, there was a couple of questions on machine learning and better analysis, quantification of this. We're all ears, because we're not, again, we haven't done this well. We need to do this much better. So happy to collaborate or discuss further, explore options.